I want to say, got it. So I just wanted to say, thank you so much for the invitation to speak. It's really an honor to speak to this group about this work. And I'm going to be speaking to you today about some work in my lab that was led by Isabel Fenton, who was my former postdoc on planktonic foraminifera. And I have to say off the bat that I know a lot about planktonic foraminifera now, but I'm certainly no expert, and I dabble in lots of different groups. And I would say that most of my expertise is in mollusks, but I've had the pleasure of working with planktonic foraminifera with Isabel and also with folks like Tracy Hayes. And the talk today centers on the emergence of the latitudinal diversity gradient in planktonic foraminifera. So as most of you will know, the latitudinal diversity gradient is one of the largest scale and longest known patterns in ecology. And it describes this pattern of increasing numbers of species observed from the poles to the equator. And I am really interested in LDGs in general because I think they can tell us a lot about the drivers of diversification on long time scales and what structures species distributions, speciation, extinction over time. And so today I'm going to talk to you about the emergence of the latitudinal diversity gradient in planktonic foraminifera. Let's see if I can advance it. So latitudinal diversity gradient was first discovered over 200 years ago by Alexander von Humboldt, when he described the increase in the variety of structure, grace of form, and mixture of colors towards the equator. And since Alexander von Humboldt first described the diversity gradient, there have been over 100 hypotheses proposed to explain why we see higher diversity in the tropics. And what I think is really exceptional about the diversity gradient is the pervasiveness of this pattern. So we find diversity gradients on land, like you see here for vertebrate diversity. We see diversity gradients in the sea, like you see here for copepod diversity. We see diversity gradients not only in animals, but also in plants, like you see here for vascular plants. And diversity gradients have been studied really intensively in both the marine realm and in the terrestrial realm today. And there's also been a lot of work done to quantify diversity gradients at specific times in Earth history. But despite all of this intensive work, there's still a lot of debate about the causes and the causal mechanisms behind LDGs. And ultimately, when thinking about causal mechanisms, they boil down to, and they have to invoke, differences in regional rates of speciation, extinction, local extirpation, and the movement of species into or out of regions. And really, these four processes are the only way to generate differential numbers of species across space. So to have differential numbers of biodiversity across latitudes. And these processes themselves are thought to be controlled by a suite of abiotic and biotic factors. So things like spatiotemporal climate change, species interactions, and available area, so land area or shallow marine area if we're talking about the sea, and resources, so things like nutrients and necessary biotic components. The problem, though, with identifying causal factors is that modern studies have a lot of difficulty quantifying rates of speciation and extinction, and this is because those processes often happen over longer timescales. And the other problem is that many of the factors that are thought to affect rates of speciation and extinction are collinear with latitude today. So you can see that here for temperature and precipitation. They sort of vary with latitude. So this makes it really difficult to disentangle which factors are actually driving higher rates of diversity at low latitudes. The fossil record, though, can actually provide us with an understanding of how rates of extinction and speciation have varied spatially over long time scales. And the fossil record, or the past more generally, can also provide us with times when the relationship between latitude and hypothesized drivers like speciation and extinction, driven by temperature, precipitation, etc., were different from today. So times when they were not collinear with latitude. And so in this way, we can actually look to the past to gain insight on which of these factors may actually be key in driving LDGs. So we decided to look to the past to gain better insight on the formation of latitudinal diversity gradients using the fossil record of planktonic foraminifera. And this is because, as all of you know very well here, planktonic foraminifera have an excellent fossil record, both spatially, but also temporally. And they're fairly well resolved taxonomically, although of course I know that issues remain. And work in my lab has been focused on creating a new database for planktonic foraminifera called Triton. And this is work led by Isabel Fenton, who is a former postdoc in my lab. And what's great about Triton is that it's now the largest data set for any fossil clade to date, and it contains more than 500,000 records over the Cenozoic. And so this is about 3 times larger than the previous largest data set for planktonic foraminifera, which was Neptune. And to put our data set again in context, the largest macrofossil data set is bivalves, and that comes from the Paleobiology Database, or PBDB. And this has roughly about 197,000 records for the entirety of the Phanerozoic, and only about 79,000 records for the Cenozoic. And with Triton, what we did was we ensured comparability among data sources that we were compiling from by cleaning the occurrence records using a unified set of taxonomic concepts. We converted all of the ages to the new GTS 2020 timescale. We also generated new age models, which were done by generalized additive models, or GAMs. And we also rotated all of the coordinates, so the present day latitude and longitude points, to their paleo positions using a unified paleo coordinate rotation system. And on average, you can see that these efforts resulted in a large increase in the number of occurrence data for each time period and each latitudinal zone or region between Neptune and Triton, which is our new database. The densest records, of course, in Triton are towards the present, which isn't at all surprising, but the records are also fairly dense across latitudes, even going as far back as the Eocene. So for any of you interested in the database, please do check it out. It's now freely available, and I'm happy to answer any questions about it as well. So using Triton, Isabel and I wanted to quantify spatial patterns in planktonic foraminifera over the last 40 million years at a higher resolution than had been possible before in the fossil record. And our goal was to potentially provide new insight on drivers for LDGs by relying on the over 400,000 unique species by locality by time records in Triton. So what we did was we quantified latitudinal diversity gradients in different ways. So we did this using percentiles derived from point occurrence richness estimates. So for example, for each of these drill holes or places on Earth, you have specific times that you can estimate richness. And then for a particular time bin, you can then take, say, the 90th, 95th or 90th or 75th percentile of richness for that time and that latitudinal band. But we can also calculate latitudinal diversity gradients with subsampling, and we used a variety of different subsampling approaches. So we could do subsampling by site and age, or by species record. And we tested a number of different spatial and temporal binning schemes to evaluate the sensitivity of the patterns based on that temporal and spatial binning scheme. So what I'll be showing you is LDGs primarily calculated or derived from centiles, so percentiles from those point level richness estimates within 2.5 million year time bins. But the results were fairly congruent regardless of the subsampling approach that we used or that LDG construction strategy. And you can see this here for the time bin centered on 1.25 million years, and then the time bin centered on 11.25 million years ago. And so this is our centile approach. So in that centile approach, as I mentioned, the point level richness estimates were binned to 15 degrees latitude. And then LDGs were constructed using the 75th percentile of the richness within each latitude by time bin. But again, choice of percentile didn't really affect the patterns much. And you can see this here for the time bin focused on 1.25 million years. You still get the overall shape, but obviously the magnitude will vary. The only percentile that did differ were like the lowest ones. And this should make a lot of sense to us because richness is low even at low latitudes in the modern. So equatorial areas can be really depopulated in species like you see here for modern fishes. So this is a diversity gradient for modern marine fishes. And you can see that even at very low latitudes, you can have quite low diversity. And we see this pattern replicated on land as well in birds and amphibians. Even at low latitudes, you can have areas in the tropics that are very depopulated in species. So it's really the upper tails of the distribution that define latitudinal diversity gradients. And I think this is important to keep in mind that diversity varies a lot, both latitudinally, but also longitudinally. All of the approaches that we use to estimate latitudinal diversity gradients revealed fairly significant changes in the spatial distribution of planktonic foraminifera over the last 40 million years. And what we found is that a modern style latitudinal diversity gradient began to emerge gradually about 34 million years ago, but that it remained fairly shallow in slope until about 15 to 10 million years ago. And you can see that here with the colors. So the colors indicate the ages, with the red being the oldest and then blue the youngest. This is the same patterns, but showing a subsampling approach. So this was a centile approach, and this is a subsampling approach. And the dark lines here represent the mean of 1,000 iterations, and the shading the 95% confidence intervals. You also might notice the gaps here, and those gaps are for our older time bins where we didn't have sufficient samples to do our subsampling approach. And in this particular case, that was 15 samples for that particular latitude by time bin. So using the diversity gradient estimates that we got previously, either through our subsampling approach or the centile approach, we could then actually quantify the gradient or the actual slope of those diversity gradients over time. And what we did was we standardized within each time period. And so if we standardize the gradients within each time period so that the maximum diversity is 1, the pattern is going to look something like this, going from the past to the present here on the left, where we get increasingly steep gradients towards the present, which isn't surprising, of course, based on the patterns that I showed you previously. And what we did to estimate these gradients was just use a linear model of richness as a function of absolute latitude. So we were collapsing both the northern and the southern hemispheres on top of each other. Again, patterns were insensitive to choice of methodology. So this is what I'm showing you here for the centile approach. So this was from subsampling, and then this is the centile approach for the northern hemisphere, the southern hemisphere, and then both hemispheres combined. And overall, these patterns support previous suggestions that shallower diversity gradients occur during warmer time periods and steeper gradients occur during cooler time periods. Okay, so once we quantified these latitudinal diversity gradients through time, once we actually got those patterns, we wanted to try to identify potential drivers of these LDG changes that we were seeing over the last 40 million years. And to do this, we used two different modeling approaches. We wanted to investigate the relationship between richness and climatic factors within time bins, but also temporally across time bins. So in the first modeling approach, we modeled richness as a function of 4 variables: mean annual sea surface salinity, mean annual thermocline extent, mean annual mixed layer depth, and mean annual sea surface temperature within 2.5 million year time bins. And to do this, we use spatial autoregressive models, and we wanted to see which of these variables were predicting patterns of richness. Then in the second modeling approach, we investigated the relationship between change in richness and then change in those same 4 climate variables at given locations on Earth. So we did this over varying intervals of time. So we looked at the change over 2.5 million year intervals, over 5 million year intervals, 7.5 million year intervals, 10 and 12.5 million year intervals of time. And the climate data that we used came from the HAD-CM3L coupled atmosphere-ocean climate model, which was in collaboration with Paul Valdez and Alex Farnsworth at the University of Bristol. And we needed to use these climate model data rather than, say, proxy data, because our model framework actually required spatially and temporally explicit estimates of climate or the environment at each of our estimates of richness. And this is just not available with proxy data for the last 40 million years for all of the locations that we had on Earth. So what did we find? Well, results from both of the types of models that we ran, so both the spatial autoregressive models within our time bins, but also the models across time bins, suggested that really sea surface temperature was the only variable that exhibited a consistent and strong positive relationship with richness over time. And again, this isn't a particularly surprising result. And this is what I'm showing you here for the spatial model. So within each of those 2.5 million year time bins. And this relationship of richness with sea surface temperature persisted for at least the last 15 million years and perhaps even longer, but the confidence intervals overlap 0 further back in time. We also wanted to include temperature range of the thermocline, so the variability in temperature that you would get in the thermocline. But actually, we couldn't do this because, again, unsurprisingly, this was highly correlated with sea surface temperature. And so you couldn't include both of those variables in the same models. But when we swapped sea surface temperature for temperature range of the thermocline, it exhibited similar patterns to what we were seeing in sea surface temperature. So it was significant and exhibited a positive relationship with richness over the last 15 million years. So both models seem to agree that temperature and temperature variability correlate positively with richness in models built both across space and also through time. And these results and a lot of previous work suggest that the changes we see in the diversity gradient for planktonic foraminifera mirror the changes we see in the steepness of the latitudinal temperature gradient. So we see initial formation of the diversity gradient beginning about 34 million years ago, coincident with the transition from warm house to cool house conditions. And then we see a significant steepening of the diversity gradient, coincident with a steepening of the latitudinal temperature gradient about 15 million years ago. And what's important is that this steepening of the latitudinal temperature gradient is associated with an increase in vertical temperature structure at low latitudes. So as cooler bottom waters are brought down from higher latitudes to the tropics. And given these patterns, we hypothesized then that the emergence of the modern day latitudinal diversity gradient in planktonic foraminifera may have been caused by enhanced thermal niche partitioning at low latitudes, and that this might have increased speciation at low latitudes or caused the collapse of communities at higher latitudes as those warmer water niches were removed. And so one way to test this hypothesis is to examine changes in the degree to which species of planktonic foraminifera partitioned by depths within the water column over time and across space. And one way that we can sort of examine this is to look at evenness. And evenness, as I'm sure all of you know, looks at how the number of species are partitioned across habitats. So in this case, across various depth habitats within the water column. So low evenness would indicate more species are present at one depth over other depths. And in this case, we examined how species partition between 3 different depths, so the mixed layer, the thermocline, and the subthermocline. And we did this across time and across latitudes. And what we found was that our hypothesis was supported by evenness changes within the water column. So low latitude assemblages of species today are more evenly distributed vertically within the water column compared to assemblages at high latitudes. Again, this is not that surprising to anyone here, but that assemblages exhibited more evenness across depths and across latitudes when the gradient was shallower millions of years ago. And so this pattern may imply that as warmer waters at high latitudes further back in time, you had these warmer waters, you could then support a broader range of vertical temperature habitats deeper in time. So you had diversity partition more evenly across those temperature habitats. But that as these warmer waters disappeared, you collapsed these communities and these species were either extirpated or went extinct. And so given this, we would expect higher extirpation or extinction at high latitudes, and we could potentially expect to have more speciation at low latitudes associated with that increased vertical temperature structure that I mentioned, as those colder bottom waters were being brought down to the tropics. And of course, both high latitude extirpation or extinction and low latitude speciation would contribute to the emergence of an LDG. And so again, we wanted to test this by looking at rates of speciation, extinction, extirpation, and dispersal in low versus high latitudes. And in this case, we define this as within or exclusive of 30 degrees latitude. And so what we found was that, and this is consistent with our expectation, is that low latitude speciation began to exceed high latitude speciation after 30 million years. And again, this is suggesting that the modern day LDG in planktonic foraminifera is being driven by this low latitude origination. And in addition to low latitude speciation, local extirpation at high latitudes is also contributing to the emergence of the LDG. But in this case, it tended to have a smaller effect than speciation. We actually didn't find any evidence that extinction steepened the diversity gradient. And this is because extinction was actually higher at low latitudes beginning about 20 million years ago. So the reverse of what you would expect. And similarly, dispersal from high to low latitudes didn't contribute to the emergence of a modern day LDG either. And this was because we see dispersal was reversed. So it occurred predominantly from low to high latitudes, so an out of the tropics pattern for at least the last 10 million years. So again, sort of what you would, the reverse of what you would expect if it were to be contributing to the emergence of the modern day LDG. So taken together, these results suggest that the modern day LDG for planktonic foraminifera is controlled potentially by speciation at low latitudes and extirpation at high latitudes over the last 30 to 15 million years. And so what we are proposing, and this is very much a hypothesis based on the patterns that we were seeing in the data, but I think it's supported by these different lines of evidence, is that the modern day LDG for planktonic foraminifera may be controlled by high latitude cooling that brought colder bottom waters down to the tropics. This increased latitudinal temperature gradients, but it also increased vertical temperature gradients at low latitudes. And these increased vertical temperature structure within the water column at low latitudes may have enhanced niche or niche partitioning there. And this might have provided more opportunities for speciation, explaining the higher rates of speciation we saw at low latitudes. And consistent with this hypothesis, the tropics today are richer than the tropics of the Eocene and the Miocene with regard to planktonic foraminifera. And this potentially has to do with this stronger vertical temperature structure that was weak to absent during these warmer time periods. The other, I think, important thing to note is that the extreme warmth during the Eocene might have also exceeded species thermal tolerances at low latitudes, which could have depressed diversity equatorially at these times. And of course, there is some concern that this might be occurring today, or at least will occur in the future. So if these conclusions hold to further scrutiny, then low latitude speciation was the primary contributor to the emergence of the modern style LDG and planktonic foraminifera, and this really began to accelerate around 15 million years ago. And what I find particularly interesting is that some of my past work has also suggested low latitude speciation contributed to LDGs on land, which is what I'm going to very briefly discuss today. And I know this is a bit of a departure from planktonic foraminifera, but I think the mechanisms are roughly similar, which is it speaks to a potentially sort of universal mechanism for why there might be higher diversity at low latitudes, at least at some points in time. So in this past work on terrestrial LDGs, I used an eco-evolutionary model to test whether climate change, so both climate change across space, but also through time, can generate latitudinal diversity gradients from no starting biodiversity pattern. And my specific hypothesis was that spatiotemporal climate change at low latitudes could drive higher rates of range fragmentation there. And this would result in speciation, and this would then pile up species in the tropics. And I used, as I said, a spatial modeling framework to get at this. And I was specifically could use virtual species. And these virtual species could move about the landscape. And this landscape was divided into grid cells. And the species could move about based on an assigned dispersal ability. And they could only occupy grid cells that were climatically suitable for them. And these climatic tolerances were also assigned. But the climate in each grid cell, or the conditions in each grid cell, could change through time, depending on how the climate was changing. And this, of course, would affect whether cells remain suitable for species. And in this case, the changing climate conditions were represented by a 120,000-year record of terrestrial climate change. And in this case, year 0 was 120,000 years ago, and then we're stepping forward into the present. And the amazing thing about this climatic record is that it was modeled every 100 years. So we have this snapshot fairly continuously in time that we can then prime the simulation with. And each point here represents a particular point or grid cell on Earth with specific temperature and precipitation conditions. And you can see how these are shifting and changing through time. And then in the simulations, change in climate across space and through time could then instigate 4 different processes that we've already been discussing in the study on planktonic foraminifera. So speciation, extinction, extirpation, and species movement or dispersal. And speciation would occur, as I said, through that range fragmentation that was instigated as a result of climate change. And if populations remained isolated for a certain amount of time, then you would see speciation result. And that's being shown here in the top panel. And then extinction would occur if all of the occupied suitable habitat for a species disappeared. So a species wasn't able to find any suitable conditions. And what we found from these simulations was that we could replicate latitudinal diversity gradients using this simulation framework. And I think that this is pretty exciting because basically all we did was place some initial points on Earth and then give those initial points a dispersal ability and some climatic tolerances, and then watched how those points or those virtual species responded to realistic estimates of climate change over the last 120,000 years. And so from no starting diversity gradient, we could actually produce a diversity pattern that looks remarkably similar to empirical diversity gradients like you see here for birds and amphibians and mammals. And this diversity gradient was formed primarily due to higher rates of speciation at low latitudes, just like we saw in the study using empirical data for planktonic foraminifera. And in this case, the spatiotemporal climate dynamics of climate near the equator, so at low latitudes, fragmented species ranges more. And this range fragmentation then jacked up rates of allopatric speciation and piled up species in the tropics. And so this could have been a similar, obviously very different, right? Because we're talking about a vertical water column structure compared to the two-dimensional space that I modeled on land. But perhaps you're getting sort of similar dynamics driven by different parameters of the climate, both across space and through time. And so maybe speciation is really, low latitude speciation is really a key driver of the diversity gradient. So hopefully I've provided some evidence that maybe we have steeper diversity gradients during cold intervals of time, that higher equatorial diversity may be driven by higher low latitude speciation, and that maybe spatiotemporal environmental heterogeneity may drive this low latitude speciation. And I think one of the things we have to be very careful about is accounting for spatial sampling biases when we're quantifying these diversity gradients. And we tried to do this very carefully using a number of different approaches in the study on planktonic foraminifera. But I think it's even more important when looking at things like shallow marine invertebrates, which is something I'm currently working on. So yeah, I just wanted to thank you again for the honors to speak. It's really exciting to talk to foraminiferal experts, and I especially want to thank all of my collaborators and my funders, and especially Isabel Fenton, who led the planktonic foraminiferal work. So thanks again.
Well, thank you so much, Erin. Excellent talk, and congratulations on this amazing database. The work you and Isabel put into this, and Adam and others, is really paying off. I mean, the kind of macroevolutionary and macroecological questions that you can answer now is amazing, and it's only going to get better as you push this back in time.
Absolutely. I definitely should mention that Adam Woodhouse co-led the Triton database, and he is now doing really important work trying to, as you said, push it further back in time and get even better records for around the Cretaceous period. So yeah.
Fabulous. If you've got questions, it'd be great if you can raise your hand using the reactions button, and if you don't want to be on camera or use the mic, you can put your questions into the chat. So anybody want to come forward and ask Erin a question about her talk? I guess I can start one off. Oh, Mikael, yes, go ahead.
It's actually not Mikael. It's
we have a whole group here, so this is Lucas speaking.
Great.
Yeah. So I don't know. Did you get my name? I'm Lucas. Thanks, Erin, for the talk. My question is about higher frequency variability of the LDG, because we see variability on glacial interglacial timescales, and as we're moving into a more or warmer world again, the changes are of course also at a very different rate, and evolution probably doesn't play a role. Do you think these mechanisms then that you suggested are timescale dependent, and where is the sweet spot where other mechanisms and speciation start playing a more important role?
Yeah, I think that's a really interesting question, and that's something that we discussed with Moriaki, who is one of the reviewers, because I think some of his work on those shorter timescales is showing that dispersal, so movement, it is really an important process and is actually changing diversity patterns across latitudes. on these shorter timescales and towards the recent. And so, yeah, I think you can have, obviously, as I was saying in the talk, speciation and extinction occur over these longer time frames. And so you might see larger overall pattern changes and diversity changes as a result of speciation and extinction, but that on shorter timescales, you can still get changes as a result potentially of more species extirpation or movement. And so it would be interesting to look at how that scaling actually works. One of the reasons that we focused on the 2.5 million year time bin interval, although we did test shorter intervals of time too. So if you're interested in those patterns, the data are provided in the supplement of the paper for at least million year intervals. I can't remember if we did even shorter, but one of the problems is that, of course, your data gets sparser as you get to finer temporal windows, and it's harder then to get sufficient numbers of records to look across latitudes, especially further back in time. So it was this kind of balance between sufficient numbers of records to feel confident in the diversity patterns, but also getting a fine enough temporal window to feel like you're actually capturing realistic diversity estimates across latitudes.
Great, thanks, Erin. Mikael, any other questions from your group?
I do have a question actually as well. So Erin, this is a great talk, a very interesting study. You were talking about the fact that if many things are co-varying, it is difficult to figure out what is actually driving here. And here in this long-term trend, the very moment when you are talking about allopatric speciation, we have the problem that at the same time as you have the change in climate throughout the Cenozoic, you also have a progressive fragmentation of the tropical oceans. And I wonder what role does that play if allopatric speciation would be the process that in your model is generating the diversity?
Yeah, I think that's a really interesting question, and it's not something that we analyzed explicitly in this study, but I think it absolutely could play a role, and it certainly fits in with sort of my worldview of allopatry driving low latitude speciation, and that's what we were finding on land at least. And so it might be sort of in addition to sort of vertical temperature structure, sort of fragmenting ranges or creating more environments, like you could also have that across space as well, in more of a 2D structure. So yeah, I think that's a really interesting idea. I think it's definitely something that would be fun to try to model but yeah, we didn't do it in this study.
Thank you.
Anybody else have a question? So Erin, last month, Adrian Lamb gave a really good talk on diachronidae among planktic foraminifera. And one of the things that really shocked me is the millions of years diachronidae going from the temperate South Pacific to subequatorial North Pacific or things like that. And so despite that, your Triton database is pulling up, the general patterns, which, tells us some really interesting things about the link with climate. One of the things that completely puzzles me is how the speciation process works in planktic foraminifera, and also stasis. How do you maintain stasis and stability for so long among the populations? And if you haven't scuba dived, but if you've ever collected living planktics, you realize just how far apart individual specimens are, and what is competition or what is incumbency. How does this work? So I'm looking to Mikael and his group and others to start solving these puzzles. But for me, as more of a deep time paleontologist, it's kind of shocking because I make certain assumptions about the timing of evolution and synchronicity of their distribution and so forth. And boy, there's just so much more complexity. And studying the modern as well as the Neogene assemblages is really critical. And this database really pulls it out with so much good time control.
Yeah, I mean, I completely agree with you. I have had many discussions about how speciation actually would work, and with planktonic foraminifera, and given exactly the constraints and parameters you just said. And Michal, I think that would be great if you to try to solve that. And it's something that has really puzzled me as well, and something that I wish I had more answers for you, but yeah.
Well, stay tuned. I think
This is why I think there's still a lot of uncertainty in sort of the mechanism. That's why it's still very much a hypothesis, right? Because like how is the speciation actually occurring?
working at it from different angles is going to be key. And so this is really great. So Daniel, you want to ask your question? You want to unmute and ask your question? Or I can ask it for you. All right, Daniel asks, my question is, given the various processes that occur at the ocean level, is the latitudinal gradient of speciation the same on both margins of the ocean?
Yeah, it's a great question. And we did actually try to tease apart these patterns by ocean basin, but you ran into a similar problem to the time bins, where you lost a lot of data and you had a lot of gappiness. And unfortunately, we didn't really feel confident about saying anything concrete about differences in terms of margins or in terms of ocean basins. That's not to say that you couldn't do it on sort of a shorter temporal window or focus on specific regions. But we did try to look into that very question, and we just, at the scale of our analyses, we didn't really feel comfortable making grand conclusions, but it's a great question.
Good. Anybody else?
No, we didn't. So, Rue, I'm sorry if I said Ryu. I'm sorry if I say it.
Right. I'm sorry.
Maybe you want to say your question. Sorry.
Yeah. So yeah, thank you, Aaron. My question is basically, about the driver of this latitudinal diversity gradient. And certainly the HCM3 has the temperature, but they miss the part of food variability. And I'm wondering if you have, at least compared to those environmental proxies, for example, biological pump or other things, carbon isotopes, and infer how is the impact of food variability.
Yeah, that's a great question. Like, as you said, HCM3, and I think, most climate models, they don't have that nutrient component to the models. And so, and there are models that can provide that nutrient component, but most of the time they are at much lower resolution and they have much larger grid cell boxes, and they're not fully coupled. One of the exciting things is that there is plans now for various groups, but particularly the Bristol group with HCM3, to run these models now with oxygen and nutrients coupled into the model, which is like very, very exciting. So you could actually go back and look at your very question.
Yeah.
So in terms of nutrients and food availability, we couldn't look at that across all of the time that we were looking at and across all the space. But one of the interesting things, right, is that as you probably all know far better than me, planktonic foraminifera have very high diversity in the gyres, and those are often very nutrient poor. So it doesn't seem like there's a very strong correspondence between nutrients and species richness in planktonic foraminifera. So that was our line in the paper. But it would be great to test it more explicitly.
Yeah, thank you.
So Erin, one of the challenges as you go back further in time is interpreting depth of the thermocline, and it becomes more and more subjective. We do vertical oxygen and carbon isotope gradients, and you can't assume morphologies as an indicator. And so what kind of independent indicators of depth of the thermocline might you be trying to work with to get a handle on that?
So what we did was we used the climate model data themselves. So we were able to actually model, so for every one of the locations on Earth where we had the fossils, we actually modeled, and for every time point, we modeled the temperature with depth, and then we generated our own with
Oh,
Paul
that
Valdez.
makes sense. Yeah, great.
Yeah.
Good. Okay, any other questions? Well, Erin, thank you for a fantastic talk. And
Thanks so much again for inviting me, and thanks everyone for being here and listening.
yeah,
Any feedback, most welcome.